The secret to protecting the nation's hog herd may lie in their genes. Randy Prather from University of Missouri Animal Sciences explains how he developed the first genetically engineered pigs for researching animal-related diseases.
If we can change the pig's genome, the pig has a 3 billion letter genome. If we can change a few letters of that genome, we can make pigs resistant to diseases like porcine reproductive and respiratory syndrome virus, transmissible gastroenteritis virus, Seneca Valley virus. Just by changing a few letters of the genome can have a huge impact. PRRS, for example, cost producers in North America, and this is decade-old information, estimated in North America to be $660 million a year. We have a solution for this disease. To genetically engineer the pigs so they're resistant to PRRS, it's really a pretty straightforward process. We can go in and do something called zygote injection. We get a freshly fertilized embryo, we inject some editing reagents, and it edits that 3 billion letter genome, and it can change just a couple letters, and that will make it so that the pig doesn't get sick. It doesn't see the virus. So there's a molecule that basically sticks up on the cell surface that the virus recognizes to infect the cell. By changing a couple letters of the genome, you can get rid of that molecule, and now the virus has no way to get in. There's lots of variations for genetic engineering, and I use the broad term because it can be as simple as changing a couple letters of the genome, a 3 billion letter genome. It can be as elaborate as going to another species and getting their gene and putting it in the pig. So for example, we've taken a gene from C. elegans, which is a little roundworm, and the gene encodes a fatty acid desaturase. We put it into pigs. The pigs make their own omega-3 fatty acids. So we can kind of cut and paste, and so that's a little bit more elaborate type of genetic modification. The very simple one is to just change a couple letters, and in the case of PRRS, get rid of CD163, and the pigs don't get sick. We've done the same thing with another protein called aminopeptidase N, another molecule that sticks up on the cell surface. In this case, transmissible gastroenteritis recognizes that molecule. You get rid of it, the pigs don't get sick.